Thanks very much to the organisers for inviting me here. It's been a really fascinating conference. What I want to do in this talk is to do three things. I want to describe really quite briefly a project that Angela and I lead in Oxford, which we call the Restatement project, in which we try and do what are a type of systematic review. I then want to explore one particular topic we've done, which interestingly has come up several times during the last few days, which is the issue of the harm and potential harm that neonicotinoid insecticides do on pollinators. And then I want to finish by suggesting some lessons, at least we think that we've taken from doing this, that might have more wider impact and relevance even for organisations such as the BES. So that's my co-author there, and that's the Oxford Martin School. What we try and do is we try and do systematic reviews, and these are very much systematic reviews which are pulled by a policy need. So they're really quite narrow systematic reviews compared with some of the ones we've heard about in the last couple of days, and they really are pulled from a specific policy need at the moment. They're very much targeted toward policymakers and other stakeholders, and they aim to be policy neutral, knowing, of course, that policy neutrality is a goal. It's something you can't completely achieve. I very much like Roger Pielka's sort of taxonomy of the way that we as scientists engage with policymakers. And he categorizes that into, I think, 4 different classes. And for the talk here, I want to distinguish between when we're acting as honest brokers, when we're trying to take some evidence and try and explicate that for a broader audience, and then when we're trying to be advocates for a particular policy. And we as individuals, we do both of those things at different times. And I think the complexity arises when they get confused. I don't know if it's Andy's term, actually, when you become stealth advocates, when you're claiming to be an honest broker, but actually you're being an advocate. So we're aiming to be honest brokers in this case. And we do the systematic review, and like other systematic reviews, we try and state where uncertainty exists. And we use the sort of classification of reserved vocabulary, which changes from study to study. I'm not going to go through it. This is an example we used in the first one we did, which was looking at issues surrounding bovine TB. So it's not a ranking of the data, but a description of the different types of data. So very briefly, what we do is we identify what we call, in this terrible jargon term, a super stakeholder, typically the group that has policy responsibility for it. For most, we've done 3 studies so far and have 3 in train, and for most cases it turns out to be a government department. We recruit an expert group, and we try and get an expert group that is broad, people who, when they're in advocacy mode, may be taking different views on a controversial topic. We draft a restatement. People are fabulous in giving up their time to be involved in a process such as this, so we get them to Oxford. We treat them as well as we possibly can, which means raiding the wine cellars, and to look after them. We produce the restatement. We then take it out to a stakeholder engagement. Typically 40 or 50 people. I think probably everyone here at some stage writes papers and things, and so you know what it's a pain when you get 2 or 3 referees' reports back. We get 30 or 40 referees' reports back, but the restatement the review is hugely improved by that process. And just looking around the audience, I can see people who have been involved in it, and we are extremely grateful for that. It then goes through another iterative revision. Everyone on the writing group has to be happy with it. I know Rosie Woodruff is at the meeting. So I remember the very first one we did with bovine TB, getting one particular very controversial sentence right. Rosie was in a Land Rover studying wild dogs, wasn't it? In a safari park in Kenya, and I was in a rainy office in Oxford, and I knew there's something terribly wrong with life choices I'd made at that point. So we managed to get that done, and then, and again, we all understand, I suspect in this audience, that the sort of peer review, the positive and things, we then have it peer reviewed separately and published in a journal, and that gives a final check on it. But it's also very important because peer review, perhaps more than it should do, does have this sort of mystical imprimatur. And so it comes out, the way we've done it so far is that there is a paper which is extremely short, almost a stub, And then the restatement itself is an appendix to that paper. And it's written in such a way that probably someone coming to it completely for the first time wouldn't be able to do it. But our target audience is a sort of senior civil servant or someone working in NGOs who has had policy responsibility for that area, but might have read classics at university or something like that. So the restatements itself is a set of paragraphs that are internally provide all the information, but then there is an annotated bibliography which can be much larger than the restatements, which provides notes and some explanation of how the evidence leads into that. And where there is controversy and disagreement, then that will explain how the writing group arrived at the restatement, and then the entry into the literature itself. So that's a process. Let me give a brief example into how we did it with one particular case. And this is the issue of neonicotinoids, insecticides, and pollinators. So we've seen massive declines in pollinators across most of the developed world. There are particular issues with honeybees, the colony collapse disorder, and in particular the spread of this horrible mite, which has gone through most of the areas where honeybees are produced, are common. Neonicotinoids are a class of insecticide. They were introduced in the 1990s. They're now over 30% of the market. There are different types of neonicotinoids, but most of the attention is on a type that are used as seed dressing. So the seed is coated by this chemical, it gets into the plant, and it moves systemically through the plant. So any insect that feeds on that plant encounters the neonicotinoids, but they also get into the pollen, they also get into the nectar, hence the problem. And after a sort of decade of increasing concern about this, the EU brought in a partial restriction in 2013, and the big policy issue for government was whether they should be supporting this policy when it came up for renewal, as it is at the moment. So we've got together a group of experts in this group. You've already seen a photograph of Simon Potts in Lynn Dix's talk. And these are people who, again, in advocacy mode, when they're in advocacy mode, have had different views about what policy in neonics should be. I'm not going to go through a great amount of the detail, but we do what might be called honeybee home economics. We work out what the typical concentration of the insecticide is going to be in the pollen or the nectar, how much bees will consume. And the result, and I think it's pretty much a consensus result among virtually everyone, is that the typical amount of neonicotinoids that a pollinator is going to be consuming is some way below what is going to be a lethal dose. So these are not insecticides that are likely to kill honeybees and other pollinators, at least when they're used properly. There are examples where they're used improperly, where this has happened. But there are so that's the simple bit, but there are a whole load of complexities that make it a really, really difficult question. Perhaps even a wicked question to use the phrase Lucy used. So the issues include, and I'm just going to go through these quite quickly, issues of chronic versus acute exposure. Most LD50s are measured as a chronic dose. In fact, most pollinators consume neonics over a long period. There is now a lot of evidence that sublethal doses of neonicotinoids, down to the type that it is likely that pollinators are going to consume quite frequently in the field, will have sublethal effects. And these sublethal effects, in particular, are behavioral effects. We know that there are possibilities of interactions with other stressors, especially diseases, to a certain extent, other insecticides as well. We know quite a bit about honeybees. We know a reasonable amount about bumblebees. We know very little about other bees, and virtually nothing about non-hymenopteran pollinators. The vast majority of the evidence that we have are from laboratory studies, and then moving into semi-field studies, where you artificially dose a bee and then look at its behavior in the field. We still have relatively little data about field studies. So those are some of the issues why it's difficult. And we produced a restatement, and I won't summarize it now because relatively soon after we did that restatement, a really important study came out, a Swiss study by Marge Rundloff, and those pictures are from her study. And the government chief scientific advisor asked us to do another restatement, even though it was only about a year after the first one, just because this was an area where the field was moving so fast. So we've actually done 2 on this specific topic. And essentially what we concluded at the time was that, as I've just said, bees frequently encounter neonics in the field at doses unlikely to kill. But the issue is, what do these sublethal effects have both to the population of pollinators in the field and also to the ecosystem services that the pollinators provide? And we summarize the data at the moment. There is evidence that, especially for bumblebees, that there was evidence coming through that there were population levels. The evidence for honeybees was far less. And we brought up some other issues, the emergence evidence of neonic buildup in the environment, and there's been more results coming on that recently. And also the issues of when you bring in the ban on neonics, what do farmers do? What do they do instead? So we published this about a year ago, and I don't know if Fiona Fox is still here, but thanks to Fiona, we did a launch at the Science Media Centre, and Fiona did her normal, wonderful job of getting a large number of environmental journalists in the room, and we got some really good response. Now, here are 2 of the headlines that came out a couple of days later, and you can actually sort of say, well, did you do any good at all when you get 2 headlines such as this? Now, actually, we were absolutely delighted by this, because if you read both The Guardian, it's Damien Carrington, who I think is a very sound journalist, and someone in Farmers Weekly. In our opinion, they got the science exactly right. And the headlines were what they put on top of the science. They integrated the effects, their worldview about the environment, their worldview about the costs of different policymakers. And these are 2 pieces of advocacy based on exactly the same amount of science. So we were, although you can sort of almost make fun of this, we're actually delighted by this. This was 2 things which were arguing about what one really needs to argue about in an area such as this, which is essentially the weightings you put onto different stakeholders, the value you accord to different outcomes, and not arguing over the science, which it should be possible to have an agreement about. So in my last 5 minutes or so, I want to explore some of the lessons that we think we've taken from this. And this is how one can improve the evidence base. So how evidence goes into policy is incredibly complex. None of us, we've all moved beyond the sort of linear model. But I suspect that there's a general agreement that however you think policy is formed or co-formed, then improving the evidence base is something that's worth thinking about. And of course we know that there are conflicts of interest, and we're pretty good, I think, at trying to identify where there are conscious conflicts of interest, financial conflicts and things. But I think we're much less good at looking at other sources of bias, especially subconscious bias. And there are other fields at the moment which are going through a period of intense self-examination, looking at the possibility of these unconscious biases and issues of replicatability. So behavioral studies in particular are really concerned about their inability to replicate results. Even fields which you would think would be more immune to this, such as cancer genetics, they're really worried about their inability to replicate studies, some of which might be due to they really are difficult experiments. But there is an increasing worry about subconscious bias. And I do like this quote from Saul Perlmutter. Saul Perlmutter is a cosmologist. He was a guy, or one of the guys who got the Nobel Prize for finding that the universe was expanding. And I like, science is an ongoing race between our inventing ways to fool ourselves and our inventing ways to avoid fooling ourselves. And I think we have to have a dialogue about subconscious bias where we recognize that it is inescapable and almost natural. And I particularly don't want, I'm going to talk about subconscious bias. I'm taking no moral high ground. I advise you not to, but if you look at my papers, they're as riven with subconscious bias as I suspect all yours are. So I really am not trying to be holier than thou in that. And there are all sorts of types of confirmation bias. We know that the way that we as humans organize our interaction with the world is through generating narratives. And that probably evolved, it's probably very good for us in the savannah to have a narrative about shaking grass is probably one of the carnivores we saw in Lucy's talk just now. But this leads to us having well understood confirmation bias. We tend to look for results that support our hypotheses. We tend to do experiments that will give us a result that will support or confirm our mental model of the process. We tend to have hypothesis myopia, as it's sometimes described, not to do different types of experiments. We tend to have disconfirmation bias, that we rationalize results that tend to go against our mental model. That's just the way we're built. Lucy, and I didn't catch who it was, one of the people who asked the question of her, talked about some of the statistical issues. And I think this is becoming an increasing problem for exactly the reasons that Lucy talked, issues of big data as well. The Texas sharpshooter fallacy, you all know this. You fire and then you do the target round where the bullets were. We have real issues with interpretation of multivariate data. P-hacking, probability hacking, where you look for the one significant result. There's a wonderful site where a statistician has collected all the phrases that have been used to describe a p-value of 0.51. It's frightening, look at it. At least I found some I'd use. My favorite one was, this result is flirting with significance. We really need to have better ways of dealing with this. And certainly in the neonicotinoid study world, there is some basically really bad statistics out there, statistics that should never have been done. And I think one of the issues that we also need to confront is badly designed incentives. So we know about financial incentives, but the publish and perish world, which, well, it's, to a certain extent, it's always been thus. Certainly in academic life, it's probably got greater. I actually think the impact agenda is really good. I support impact being part of the REF exercise, but it has to be done so carefully so that you are not incentivizing people to, incentivizing science to be impactful for the sake of impactful. So how do we get science with impact where it's general impact? And we don't talk much about it. Perhaps we don't talk about it enough in the British Ecological Society. But competition between journals, especially in ages of bibliometrics, there's enormous pressure on journals to publish exciting science and things. And is that an incentive for doing the best science? So this is my final slide. And I'm not going to go through it in detail, but I think we have to, I really like to advocate for a greater controversy, a greater conversation about how we can get this process better. Now, you could argue, and this is going back to Hugh Possingham's talk yesterday, that there are some circumstances where one shouldn't bother. I mean, again, there's always been statistical bias. So maybe, especially on the more fundamental types of science, we should just get on with it, and science is ultimately self-correcting. And that may be true on some of the more fundamental sciences, but some of the more applied sciences that are directly trying to influence policy, I don't think we can get away with that. That we are undermining our credibility with policymakers, many of whom generally want to make evidence-based or evidence-informed policy making. So there are a variety of things up there. I'm not going to go through all of them. I want to perhaps concentrate on one in particular, and I do that because this meeting is co-sponsored by the British Ecological Society, and the BES publishes some of the very best journals, sorry Rob, the best journals in ecology in the country. What can the BES, which punches above its weight because of its role as a publisher, do to try and prove this? So I think the single thing it could do that would be most important is to adopt what has been advocated in other fields, but as far as I'm aware, not very much in our field, and that is pre-experimental peer review. So you're going to do an experiment, for example, in neonics. One of the experiments, for example, big experiment being done at CEH at the moment. Now, hopefully, that is going to get into Nature or Science or even the Journal of Animal Ecology. And I want to get away for any perverse incentive to write up the paper in such a way it's more likely to get to the journal. Surely the thing to do is that the study should be submitted before it's done. It should be submitted saying exactly what the experimental design is going to be, exactly what the interpretation of that data is going to be, conditional on what the results will be. Surely it should be refereed at that moment, then later on, where there's all the temptation for post facto rationalization. Now, if you actually completely screw up the experiment, you fail to plant the sites or something, then clearly it should be rejected. But if you carry the paper through, then surely it should be published. And the old excuse was, as this will mean that journals are full up of papers which are not exciting. That doesn't matter in an electronic age. You know, if you were Nature or Science, then if it was an important study, but that turned out not to be very exciting, then publish it as a paragraph with the full paper online. So my challenge is for the BES to think very seriously about, especially Journal of Applied Ecology perhaps, how they could do innovations in this field that I think would improve the science evidence base. Thanks, Rosie.
Fantastic challenge there, thank you. Time for some quick questions. More challenges, right at the back.
Pete Carey from various bits around Cambridgeshire. I was wondering, with the restatements on such a fast-moving subject as the neonicotinoids, how you decide when to stop. So you know there's another paper coming. How can you get around that, especially if you know it's going to be influential? Do you have to have a guillotine for yourselves, or how do you do it?
Yeah, I mean, we just have the project finishes and then we do it. But I mean, your general point is really interesting, and again, it goes back to some of the issues that Lynn Dix was bringing up in her presentation yesterday. You could do this in real time, and you could do that. You could do it so the restatement was updated as new information came in. But that would be substantial. That would be costly, and then you need a type of Hugh Possingham analysis, I think, to tell you whether the benefits of doing that are worth the investment in the resources.
Is that on? Yeah. Humphrey Crick from Natural England. I was just wondering, two quick questions. One was, how does this process differ from the systematic review process that Andrew Pullen was talking about yesterday? And the other one is how do you go about deciding which topics to tackle? Have you got a panel which looks at it, or is it just a Mark Walput derived process, or how do you decide those?
So it's part of a continuum. There are different types of systematic review. Ours is a type of systematic review. I think where it differs from some of the wonderful things that Andrew has done, and this goes on to your second point, is that it is not us deciding what to do. Well, we did the very first one, and then the word got round that these were useful. So we're in a position that we have policymakers, largely from government, asking us if we can do things, and then we sort of try and work out which is the one that we can add greatest value to at the time. So, I mean, in a sense, we're doing an experiment, so it's not as if we've got a very careful logic of how to do it.
Hi, Clive Mitchell from Scottish Natural Heritage. Thank you for an excellent presentation. I read the update paper on the neonics yesterday in the workshop, which was excellent. Thank you very much for that as well. One of the things that struck me, I think, as you brought out in the presentation, was the continuing lack of certainty and need for more evidence on whether to act and how to act or not to act on the use of neonicotinoids in the farmed environment. And to my mind, clearly, some interest groups, the pesticides and so on, would take that as a basis for continuing to use neonicotinoids in the farming environment. And other people, another set of interest groups, interest against that, might say that's a basis for the advocation of the precautionary principle, and that we should stop using them until we have better evidence. And I just wondered, because the precautionary principle was, for me, conspicuous by its absence in your update, and I wondered if you could comment on the use of the precautionary principle in handling the degrees of uncertainty associated with an issue like the use of neonicotinoid pesticides.
So what we attempt to do in the restatements is, again, to set out the data as it exists and to set it out in as policy neutral way as we can, realizing, of course, that you can never be wholly policy neutral. I think you're exactly right that the decision at the moment whether you ban or you don't ban neonicotinoids, it's far from determined by the science, and it depends how you interpret that science. And in particular how you interpret the precautionary principle, which isn't a simple algorithmic rule, it's a vast number of different concepts used in different ways by many different people. And I think our aim is that we should have the discussion at the level of what do we mean by the precautionary principle, how should we weight the economic harm to farmers, should they exist, of banning neonics versus the benefits to the ecosystem services from pollinators, should they exist. We should be having the argument at that real level where the decision should be made, rather than arguing about LD50s or Bombus terrestris and things like that, where we should be able to summarize the data. Does that answer your question?
Yes, I think so. I mean, I think a key point that you just mentioned earlier is the consequences of acting or not acting on the different interest groups involved.
Yes.
Apparently we have the shortest question we've actually heard so far in the conference, and then I'm afraid we're going to have to move on, because, yep.
Sorry, hello, Rob York again. Thank you, Charles. I've heard you speak...
Where are you? Oh, there you are.
Over here. I've heard you speak about this to farmers. Why can't we mix up the audiences between who's here and who's at the for farming, and who should do that?
Well, I think you should, Rob. I mean, you...
Brilliant. Thank you for that answer. Thank you very much.